Prova. 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 Everything is fine.
Okay.
I will take some picture and then I will leave because I have some.
Of course, no worries.
Thank you so
Thank
much.
you.
GTF, that's the pharmacy that works on pancreas.
Oh, nice.
But I don't know. I know today. Okay, you can have a meeting. Okay, but from this moment we are in
Okay.
each word.
Okay. We need to take care of it. Yeah.
Siamo silenzio e presento la professoressa. Allora, buongiorno a tutti, grazie di essere venuti qua per questo seminario che sarà presentato dalla professoressa Alexandra Buha Dordevic della Facoltà di Farmacia dell'Università di Belgrado in Serbia. La professoressa Buha diciamo, è un vecchio amica della nostra università, abbiamo già numerose collaborazioni, collabora sia con me che col Dipartimento su varie tematiche, carcinoma del pancreas, metalli pesanti, è studiosa nel campo della tossicologia, ma collegata anche ai tumori. È qua come visiting professor per questa settimana e terrà due seminari, uno oggi e un altro giovedì pomeriggio. Il seminario di oggi sarà fatto in inglese ma molto lento, parlerà naturalmente delle problematiche dell'ambiente, dell'influenza dell'ambiente sullo sviluppo del carcinoma del pancreas. Sappiamo tutti che il carcinoma del pancreas è uno dei tumori, diciamo, più pericolosi per il semplice fatto che normalmente quando viene scoperto è già avanti. E quindi poter fare opera di prevenzione o capire quali sono le problematiche del carcinoma del pancreas e come poter in alcun modo prevenire o evitare è importantissimo. Quindi adesso vi chiedo il silenzio, poi alla fine la conferenza durerà una cinquantina di minuti, about 50 minutes, right?
Yeah, more or less.
40-50 minutes, poi alla fine per fare tutte le domande. Vi ringrazio e do la parola alla professoressa Alexandra Buha.
Grazie, buongiorno a tutti. So I am learning Italian, and as far as I know, this is broadcasting live, so I won't embarrass myself anymore, and I will speak in English from now on. So thank you everybody for coming to this lecture today. I hope it will be interesting for you and inspiring, and maybe prompt you to study and learn more about this topic. So today I will be speaking about pancreatic cancer development and pancreatic health in general. And we will see what are the influences that are in a way responsible for determining whether our pancreatic health will be stable or something will happen, eventually even cancer. So for the very beginning, I would like to hear from you, and I would like to ask you to scan this QR code and to answer one simple question that I have prepared for you. Now, yes. Yes, yes. Have you managed to scan the code? Is everything working? So if you have managed to do it, I will transfer it to the... So everybody joined? Now we are going to change the picture. So here is the question. So what do you think that determines our health? Is it genetic or is it environment? Let's see. Your thoughts. Okay, so thank you. As I can see, a majority of you has voted, and environment has won. And I think you are a little bit biased because you know that I'm going to talk about environment. But let's see, the truth is always somewhere between. So, to be honest, I was expecting that you would say genetics, because in most of the time when I doing this type of question, majority of people say that they believe that the health is detrimented by genetics. And in a way, I think as a human race, it is easier for us to believe that it is determined completely by genetics. It's in a way when we, when something happened and we say it's a destiny, so we couldn't avoid it, we don't have anything to do with it, it was a destiny. So same with health. If we connect health entirely to genetics, then it's not our responsibility anymore. But the truth is that in previous years, genome was really expensive. It was really studied a lot, and the majority of genome was read. And scientists were very surprised to find out that only 60% of diseases can be totally contributed to genome. So this was a great surprise because scientists believed that this is a much higher percentage. So these results were consistent with the estimate that 70% to 90% of the disease are basically environmental based. So in a way, you were right. But we need to be completely correct and say that our health is basically the product of both genome and environment. They do interact and they do have influence on each other. So you all probably have some grandparent who was smoking entire life, like for 60 years, and he managed to live up to 90 years. So this has to have some connection with a good genome. So when the scientists realized that genome is not the main determinant of our health, there was a new term that was coined, and the term was exposome. Have you heard about this term prior to this lecture? Okay, so the exposome comprises the totality of exposure to which an individual is subjected from the very conception until the death. So it includes environmental agents to which we are exposed to. It includes our socioeconomic conditions, lifestyle, diet, exercising, so everything we do during our lives and all factors to which we are exposed to. And this is the person, his name is Christopher Wilde, and he managed to come up with this term and with this entire paradigm. And the story about Christopher Wilde is very interesting because he basically introduced this term in 2005. This was the year when we still believed that the genome is everything and that nothing else is as much as important as genetics. So he tried to publish this paper containing the exposome paradigm, and he was rejected in more than 20 journals. At the end, he published this as an editorial in a journal in which he was an editor. So at the end, he needed to do something to publish it somewhere. And as you can see, from 2005 to 2010, nothing was happening. So it was just an editorial. Nobody actually paid any attention to the term exposome. But in 2010, EPA, Environmental Protection Agency, somebody from EPA noticed this editorial, and they really believed it's a good paradigm, and the things have started. So you can see the number of citations is growing, has been growing ever since. So what we do now, and what is in a way the most correct way to deal with both genome and exposome is this sentence. So now we know that genetics load the gun, but the environment pulls the trigger. And I think this is the best description of our health. So both genetics and both environment. And one very important and distinctive difference between genome and exposome is this. We cannot choose our genome and we cannot change it. We get it at the moment of our conception and we need to live with it. But when it comes to exposome, we can do many things about it. We can change it. So if we think about it, exposome is something which is very important for the prevention, and we need to study more, and we need to know more about exposome in order to make us more healthier as a population. Therefore, the research in the field of exposome is of high importance because we can change exposome in a way. So let's talk a little bit about cancer now. I'm sure that all of you know that cancer today is a global threat. And if we look at the statistics, we can see that these numbers are rising by year. Now we know that cancers are predominantly caused by environmental and behavior risk factors. Behavior risk factors, for example, smoking, we know for sure that this is not good and that smoking can produce cancer. Also, excessive use of alcohol as well. So these are all well-known carcinogenic risk factors. We also know that very important things when it comes to cancer are prevention and early detection. That's something that I think everybody knows. And also we need to make treatment and care of cancer much better. So in order to do all this, in order to enhance prevention, treatment, and care, we need to know more about cancer. We need to know more about etiology of each cancer and the pathogenesis in order to be able to identify biomarkers of early detection, to identify risk factors, and then define good prevention measures. And at the end of the day, if we know the pathogenesis, we can also prepare better treatment and care for the patients. I will shortly speak about chemical carcinogens. So these are the primary regions in the etiology of cancers, and there is this association, which I'm sure you have heard about. It's the International Agency for Cancer Research, and they're examining each chemical which is on the market, and they are trying to figure out whether this chemical is carcinogenic or not. So a majority of chemicals are classified to these 4 groups. And in group 1, we have 121 agents. What does this mean? It means that we are basically sure that these agents are carcinogenic because we have a lot of proofs from epidemiological studies. How do we have epidemiological studies with such type of agents? It's mainly epidemiological studies from people who are occupationally exposed to these agents. And then they are comparing the rate of cancer in this population with the general population who is not occupationally exposed to this agent. And if there are significant differences, then we can, with a lot of we can say that these agents are indeed carcinogenic to humans. Group 2A and 2B, we have a lot of evidences in animals, in vitro studies, that these chemicals can produce cancer. There are also evidences from human studies, but these evidences are not as strong as for the group 1. And when it comes to group 3, this is still not classified. So it doesn't mean that they are carcinogenic or they are not carcinogenic. They're just still haven't been classified. So I will now start talk about toxic metals. This is the field that I have been working on for more than 10 years. These toxic metals are very persistent environmental contaminants. They are all around us, and they are not produced by humans, and we cannot destroy them. So they are naturally occurring in our environment, but due to anthropogenic activities such as mining and industry, they are present in our environment more and more. So nowadays we have a serious problem with the exposure to general population, and how can we as a general population be exposed to toxic metals? There are 2 main types of exposure. For smokers, of course, it's smoking cigarettes, because in cigarettes you can basically find all these toxic metals. And for non-smoker, the most and the main type of exposure is through diet. So when you eat fish, when you eat leafy vegetables, when you eat meat, you are basically ingesting some of these toxic metals on a daily basis. These are some of the most important ones. So cadmium, arsenic, lead, mercury, chromium, and nickel. and they have been on the priority substance list for many agents. This basically means that they are important both in terms of their toxicity and in terms of their in terms of the fact that people are exposed to them on a daily basis. And as you can see, these are some of the most important ways of exposure to these metals. So you can see the drinking water. Some countries have problem with arsenic in drinking water. Also, you can see these industries that are polluting air. So when we breathe our air, we are indeed ingesting these toxic metals, especially with PM particulates, because in these PM particulates you can also find toxic metals. Of course, smoking, we were talking about this. Food, various types of food. I will tell you, for example, when it comes to cadmium, that there is this new study that showed that, for example, vegetarian people who are practicing vegetarian diet have higher levels of cadmium than smokers. That was a really staggering fact, but it's in a way it is logical, because in the leafy vegetables you can find a lot of cadmium because it bioaccumulates in such type of vegetables. So in a way our diet should be we should use all type of foods in our diets. In that way we will avoid accumulating certain types of toxic metals. So how can these toxic metals produce cancer? Some of these toxic metals are mutagenic, but not all of them. Others are producing oxidative stress, so they do induce oxidative stress, and oxidative stress in a way is the first step toward the carcinogenesis. Some of them are also inducing epigenetic changes, which basically means that if a mother is exposed to toxic metals during the pregnancy, her grandchildren will also be exposed to the effects of toxic metals. And at the end, mitochondrial toxicity, this is something that has also been mentioned many times in relation with toxicity of toxic metals. I mentioned a couple of times cadmium. This is a metal that I have been working mostly on. So the sources are, as I have told you, smoking, tobacco smoking, and diet. And this toxic metal has been among 10 chemicals that cause the greatest concern for human health for many years. And now we are very aware that this metal can produce toxic effects to many organs. International Agency for Cancer Research classified cadmium as a group 1 human carcinogen, which basically means that we are pretty sure that it can produce cancer, and it was classified as it was classified in group 1 based on the fact that it produced lung cancer. But many studies have shown that cadmium has a role in onset of some other cancers, such as prostate cancer, breast cancer, and pancreatic cancer. And we are going to speak more about pancreatic cancer today. And also, as you can see here, seafood and rice, these are some of the very important sources of cadmium, plus leafy vegetables, as already mentioned. So let's see a little bit about the pancreatic cancer. So this is not a very frequent cancer, but we can say that it is the most deadly ones, because it is a very important cause of cancer-related mortalities worldwide. As you can see, for the PDAC, which is a pancreatic dual adenocarcinoma, 5-year survival rate is still less than 9%. So I think that next year, previous year, they managed to make it 10%, but this is still very, very low. The main problem with pancreatic cancer is that diagnostic biomarkers for early detections are still unknown. And the other thing is that you basically don't have any symptoms until the 4th stage, when it is too late. So it is very important to try to completely understand the etiology of pancreatic cancer and to define these diagnostic biomarkers for early detection, because as you I'm sure know, if you manage to find it in early stage, it will be curable. These data, 5-year survival rate less than 9% is when the pancreatic cancer is diagnosed at the third or fourth stage. So let's see what is the connection between cadmium exposure and pancreatic cancer. And this is something that my research group has been working for almost a decade. So the first thing that you can notice is that many of well-established risk factors for pancreatic cancer are connected with cadmium body burden. So age, smoking, etc. This is all connected with higher cadmium levels. So in recent years, there have been extensive investigations in this field to see whether there is a connection between cadmium exposure, and if there is, what are the basic mechanisms. So in the literature, you will find the conflicting data, but more evidence are pointing to the fact that cadmium exposure to cadmium can be one of the risk factors for pancreatic cancer development. So I will show you some of our results. So this was the first study that we have done, and this was a human case control study, which was carried out at the Clinical Center of Serbia, Belgrade. So we had cases, these were 31 newly diagnosed patients with PDA, and according to the clinical situation, they have undergone Whipple procedure or total pancreatomy, which is something that is usually happening when a person is diagnosed with pancreatic cancer. And for each patient, surgeons removed 2 specimens of pancreatic tissue. One was taken from the cancerous tissue, and the other sample was taken from the surrounding non-cancerous tissue. And as controls, we used the post-mortem samples of 29 persons who accidentally died, so there were no any cancers involved in their death. And we measured the levels of cadmium in all these collected samples. So these are the results. As you can see, the levels in cancer tissue were significantly higher than the levels in controls. And one thing that was also very interesting is that the level of cadmium in surrounding non-cancer tissue was also significantly higher than the levels of cadmium in controls. And the levels of cadmium in cancer tissue were higher than the levels in surrounding non-cancerous tissue. So these were very interesting results to start with. So we basically proved that cadmium is accumulating in the cancerous tissue, but we weren't sure whether it is because it's cancerous tissue, so it's accumulating more cadmium, or the cadmium was accumulated and produced in a way onset of pancreatic cancer. So of course we continue with the examination, and here is just to show you the distributions. We also did the odds ratio and basically proved that if you are in a quartile with more cadmium levels in pancreatic tissues, your risk factors for getting pancreatic cancer are higher. Then we switched to animals because my research group is mainly experimental toxicology, so we are working mainly with animals. So we treated our rats for 24 hours with orally with high doses of cadmium. We just wanted to see whether cadmium is accumulating in pancreatic tissue. So as we have found out, it does. So lots of cadmium went to pancreas, and we with this step, we proved that even in a healthy organisms, so these rats were healthy, cadmium is going to accumulate in pancreas. So it's going to accumulate in pancreas, but is it going to do anything there? Or is it just going to be accumulated there? So the next step, in order to see what is happening in pancreas, if there is cadmium present there, was to do the in vitro study. So this was the third step, and we used two different lines of cadmium. This was done in University of Austin in USA. So they used the human pancreatic nesting expressing cell line, which are basically normal pancreatic cells, and they used the another pancreatic tumor cell lines, which represented the cancerous pancreatic tissues. And what we did is that we transformed the levels of cadmium that we measured in pancreas of our patients into the levels of cadmium that we treated the tissue. So we wanted to see what is happening in the pancreatic tissue when the levels of cadmium are the same as the levels we measured in our cases. So we used these two concentrations, and these were the tests that we have performed. And what we find that in these concentrations, the formation of free radicals after exposure to cadmium was higher, as you can see in these two concentration, and also that the caspase activity was different, pointing out to the changes in apoptosis, which can be one of the reasons why cancer has developed. So the findings suggested that cadmium could disrupt the intrinsic apoptotic pathways, leading to impaired cell death mechanisms in pancreatic cells, particularly in cancerous cells. Overall, this in vitro part of the study demonstrated that cadmium exposure does affect pancreatic cells by altering caspase activity and increasing oxidative stress, and these both activities can be introduction to carcinogenesis. So in a way, we formed this, we started from people to animals and then in vitro test and managed to prove that these levels of cadmium can produce certain changes in pancreas that in fact can lead to carcinogenesis. So these were the final conclusions from this big study. So the cadmium is indeed represented in higher concentration in cancerous pancreatic tissue. Cadmium does deposit in pancreatic tissue, even in an intact, healthy animal. And cadmium can induce oxidative stress and reduce the cell ability to enter apoptosis, which of course is an increase in tumorigenic cell formation. So the study presented three different lines of evidence, all pointing towards cadmium as an agent responsible for the development of pancreatic cancer. But we haven't stopped there. We were wondering what is happening with cadmium as a mitochondrial toxicant, and also whether cadmium can have any epigenetic effects. So we continued with the investigation, and first we also performed another in vitro study. This was also performed at the University of University of Tulsa, in Austin, USA. So the same two cells lines were used, and we used the same concentration we already established in previous study. And the main findings was that we managed to identify mitochondrial toxicity. So mitochondrial can also be a critical target in cadmium-induced pancreatic carcinogenesis. So this is one more target to think about. And these are some of the other papers we have also established. We have the collaboration with the University of Westminster, and they were also investigating the role of cadmium on microRNAs, and they find some interactions between these as well. And we also have the collaboration with Roberto and the University of Sassari, and working a lot in this field, trying to identify all the carcinogenetic pathways, because as I have already told you, if we are able to completely understand the etiology of one disease, we can easily identify early biomarkers, and by doing so, we can basically be able to diagnose pancreatic cancer much earlier, and then the success rate would be much higher. I will also tell you about the other study that my research group is still conducting. This is the DECODE EXPO study, which is basically the acronym from decoding the role of exposome in endocrine health. Because as you know, pancreas has exocrine function, but it also has endocrine function. So we wanted to see how these metals are affecting the endocrine function on pancreas, what is happening on this level. So this was the project workflow. What we basically wanted to do is we wanted to see the levels of all these metals that I was talking to you about in our general population. We wanted to see how much cadmium, arsenic, chromium, lead, mercury we have in our bodies. So the general population, non-occupationally exposed to these toxic metals. And then we transferred these doses into the doses of to treat animals with. Why we did this in this way? Because in toxicology, when we are working on toxicity studies, we are using very high doses because we want to see the effect. Because when you do an experiment and there is no effect, everybody is sad. Like, why did I do this? Nothing has happened. Everything is same. We don't have any results. Nobody is going to publish this. So therefore, we are using very high doses. But in real life conditions, we are not exposed to such high doses. We are exposed to low doses, but we are exposed to mixtures. So in order to see what is really happening in our organism, we wanted to make a real-life mixture of toxic chemicals. So we exposed our animals to the doses that our Serbian general population is exposed. And we wanted to see what is going to happen with endocrine organs, among them pancreas as well. So I will briefly show you. So I already kind of explained you all this, what we have done. And we got very interesting results when it comes to arsenic exposure and insulin production. One thing that I also forgot to tell you is the fact that we have, like for years, a problem with gender bias. So most of the time, for toxicological studies, we are using male rats. And we are translating these data to women as well. The problem is that our body is different. We are metabolizing differently. We are exposed to different chemicals in a different way. Like imagine women are spending more time in kitchen. They are spending more time in toilet during the cleaning, doing the cleaning. So we are not exposed in the same way to the same chemicals. And this also needs to be taken account. So therefore, we divided our population to men and women, and we find completely different levels of toxic metals in men and women. So men were, for example, more exposed to lead, but women were more exposed to arsenic, for example. And then we had male rats and we had female rats. And we treated them differently with different doses. Like we treated male rats with doses of a real life mixture that we obtained from our male participants, and female rats with real life mixture that we have obtained from our female participants. So we got different results for male and females. So as you can see here, this is the arsenic levels. Arsenic levels in women are much higher, 10 times higher. This is still under, let's say, allowed concentrations, because they say that everything beyond 50 micrograms per liter is still okay. It's not supposed to have any effects on the organism, but this is a very interesting result. So if you are treating female and male rats with same doses, you're not doing it right, because in reality, we are not exposed to the same doses. Therefore, our doses for arsenic in this real-life mixture were different for female and male rats. So this is what we got. Some studies have shown that arsenic has the potential to disrupt the body tolerance to glucose, to increase insulin resistance and reduce insulin secretion. But this has still been investigated. So these were the studies that already existed in the moment that we have done our study. So we have done model averaging. I won't bother you with this, but in a way, this is these two intervals that you see for men and women. This is the concentration of arsenic. So the concentration of arsenic that makes our risk to have our insulin outside the reference level for 5% is somewhere here. So for example, if you have 5.27 micrograms arsenic per liter and you're a man, you have 5% higher risk of having your insulin outside the reference dose level. So this is the interval that we got for women. And as you can see, and maybe you remember from the previous slide, so the levels for men were 0.9, and this is not in this, interval. So in a way, when it comes to arsenic, men are safe. But for women, it was 10.4. So it was even higher than this interval. So we basically find out that women in Serbia are exposed to arsenic that is increasing their risk of having their insulin outside the reference dose for 5%. And this was very concerning data. Of course, as an experimental toxicologist, we needed to check this with animals again. So we performed one more animal study. This time we treated animals for 90 days, and we made these real-life mixtures. So, and did a bunch of biochemical assessment. We did histopathology assessment of pancreas itself. We measured oxidative stress parameters in pancreas, and we did the metal measurements. This is the way the entire experiment was designed. And we wanted to see the sex differences, the differences between doses, because we had 4 types of doses. So the first one was median doses measured in our body, transferred into animal doses. Then we had the doses which were a little bit higher to correspond to people who are more exposed to these metals, because you always have this 95th percentile of people who are more exposed. We had controls, of course, and we had the doses which were reference doses. So for each of these metals, you have doses which are reference doses, and which are supposed to be safe and are regarded as safe. And these were our conclusions. So real-life exposure, we demonstrated robust evidence linking toxic metal mixtures to disrupted pancreatic function. This was our conclusions. We also managed to see sex-specific effects. So for male rats in a histopathology examination of pancreas, we see different changes than in female rats. But male rats managed to develop adaptive response to oxidative stress over time, and they managed to heal. Whereas in female rats, the oxidative stress was persistent. Also, histopathological changes were, as expected, dose and duration dependent. And since we treated rats for 19 days, and this is something that corresponds to our subchronic exposure, for example, up to 6 months exposure, we managed to find progressive damage in pancreatic tissue. So at the end, we concluded that findings emphasized the sex-specific, dose-dependent, and progressive impact of toxic metalloids exposure on pancreatic health. And of course, we concluded that we need further research to see what is happening and why we managed to find all these differences. So these would be the final remarks for all these studies that we have managed to do so far. And they say that evidence from human, animal, and in vitro studies consistently showed that cadmium accumulates in pancreatic tissue and that this accumulation correlates with oxidative stress and impaired apoptosis mechanisms, indicating cadmium's significant role in pancreatic carcinogenesis. We also find that toxic metalloid mixtures induce histopathological changes in the pancreas, such as vascular congestion, connective tissue alteration, and structural disruption of Langerhans islets. Male subject exhibited greater metabolic and oxidative stress adaptation, while female displayed prolonged oxidative damage, highlighting sex-specific responses. We also find out that exposure to toxic metals, even at environmental levels, which is something that we did with this last study, disrupts glucose regulation, enzyme activity, and mineral homeostasis. And these disruption may co-occur with pancreatic tissue accumulation of toxic metals, suggesting a direct link between environmental exposure and pancreatic dysfunction, and of course, at the end of the day, cancer development as well. So having in mind all these important things that you have learned about toxic metals and the importance of toxic metals in our health, I will just briefly remind you that we have International Society for Research on Cadmium and Trace Element Toxicity. Professor Roberto, sitting here, is the president. And our mission is to promote research in this field and also to disseminate this type of information and the type of information which is directly to general population in order to reduce exposure to these toxic metals. As I have told you, we can influence our exposome and we can influence our exposure to different environmental agents. So we can do something about this exposure. And also we have many activities such as conferences, seminars, summer school, membership opportunities. So stay tuned. This is the website of our society, and it would be really nice to join us. Thank you very much for your attention.
Ringrazio la professoressa Alexandra Buha Dordevic per la sua brillante conferenza, in cui, come avete visto, si parla sempre di più del rapporto tra metalli pesanti. Abbiamo parlato di cadmio, abbiamo parlato di arsenico, con vari tipi di patologie, carcinoma del pancreas, insulina. Si sta vedendo sempre di più che i metalli influiscono, potendo dare tumori, potendo dare collegate con malattie neurodegenerative, sono collegate con l'alimentazione, con la dieta, col mondo dell'agricoltura, col mondo economico. Quindi è una tematica che a livello mondiale si sta sviluppando sempre maggiormente in questi anni e bisognerà sempre avere un occhio di riguardo, andare a vedere le concentrazioni e le possibili ricadute negative dei metalli pesanti, ma cromo, molibdeno e altri metalli, soprattutto sull'ambiente, ma soprattutto alla nostra salute. Quindi è una tematica di grande attualità. Niente, se c'è qualche domanda che vuole essere fatta. Qualcuno?
I found it especially interesting how higher arsenic concentration lead to higher insulin resistance, and consider the burden that type 2 diabetes is on healthcare. What are there any populations with higher arsenic concentration, or maybe what are the risk factors for higher arsenic accumulation?
Particularly in Serbia, we have problem in this part of Serbia, in a city called Zrenjanin. We have like naturally higher levels of arsenic in water. So probably this is one of the reasons. Also, female body is different, of course, than male body, and probably some pharmacokinetics things are happening there that promotes, in a way, longer accumulation of arsenic in body of females. And also, if you think about it, this condition, which is called insulin resistance, it is much more frequent in women. There are some ways in which we can reduce the level of arsenic, and I will tell you one thing that is something that you can apply right now. One of the very important sources of arsenic is rice. So because of the way it is planted, and so the advice would be to use 10 times higher levels of water than the rice when you boil rice, and then you just remove this water. By doing so, lots of arsenic will be transferred into the water, and you will get the rice with much less content of arsenic. So as I have told you, we can influence our exposome, and there are things that we can do to reduce the levels of cadmium, of various chemicals. But the best way to reduce the levels of chemicals is to have better regulations, of course, and this needs to be done globally. But while we are waiting for this, there are some things that we can do on our own. Thank you.
Congratulations for your brilliant effort.
Thank you.
I have just a small question. If maybe it should be a target for the future, I don't know, but I didn't know if you make a difference between esophageal and endocrine cancer, finding metal level on your study.
No, but this is something that we plan, and we are also planning to work on IPMN, which is like another type of cancer, which is not been studied yet, and we are still not sure what are the risk factors, and this is the type of cancer that can be treated. So you are in a way following these patients for many years, and this is very important to see the risk factors, whether they are mainly genetics or there are environmental factors as well there. We didn't compare insulinomas with adenocarcinoma, but we did start to see what are the effects on the insulin production. In the future, we are planning to make like 3 different groups, like adenocarcinoma, insulinoma, and IPMN, and we will see what are the most important risk factors and how to treat them. Yeah. Thank you.
Grazie. Sono altre domande? Ok, se non ci sono altre domande, ringrazio ancora la professoressa.
Grazie.
Grazie a tutti voi.
Grazie, arrivederci.